We're really excited to present the fourth in our Medicine Informing Novel Discoveries events. And, everything is usually from a stem cell or regenerative medicine angle when we think about various diseases. Today's is really about a real labor of love. When you have somebody that cares so much about what they're doing, they apply their whole life, their whole soul to this. So we'll be talking about phage and the regenerative medicine approaches to infectious diseases. We'll hear first from Dr. Stephanie Strathsey, who will talk about her very exciting and innovative phage approach to treating really very difficult to treat bacterial infections together with Chikungunya. We'll talk with the recipient of the phage, and phage are basically viruses that invade bacteria, and you'll hear a lot more about that, but they're the perfect predator, as Stephanie will tell you a little bit more about. And then we'll hear about something I never thought would happen, an attempted cure strategy for HIV. So again, Dr. Schooley is the glue here because he's brought everybody together as somebody who ran infectious diseases and really insisted on global health initiatives together with Stephanie. But Sheldon Morris will talk about the HIV CAR T-cell trial, where we're up to, I think, the fourth patient today through the Sanford Stem Cell Institute, with Chip Schooley having helped to develop that therapy. So we've got the best in the world here, just so you know. And actually, it's not just the best, it's the only. So we want to have other people take on this approach. Here, we're undaunted by difficulty. We don't overthink things, we get things done. It's not to say we don't think about it, but we think about the most clever approaches collaboratively to get spectacular science to the clinic. So I'd like to start by having our friend Ria, who's been at 3 of these events so far, introduce the day. So we'll let Ria go ahead. This is thanks to Lee Stein and Shuba and others who really help us learn from AI. It's not just AI is learning from us, we're learning from AI. So we're going to let Ria do the introduction.
Good evening, everyone. I am Ria, an AI-enabled humanoid, and I am thrilled to be here with you tonight. Some of you may recognize me from past collaborations with the UC San Diego Sanford Stem Cell Institute, and it's wonderful to see some familiar faces in the audience. By now, I imagine you've already been part of some fascinating discussions, and I hope you're looking forward to what's next on the agenda. This evening, we'll dive into pressing topics such as antibiotic-resistant pathogens, global epidemics, and the future of infectious disease treatments. The intersection of infectious disease research and stem cell science will be highlighted, showcasing the Sanford Stem Cell Institute's groundbreaking work in translational medicine and regenerative therapies. Our esteemed UC San Diego experts will share their collaborative strategies, combining cutting-edge research and clinical trials to drive transformative breakthroughs in global health. As we face new challenges, UCSD stands at the forefront of innovation, helping us prepare for future medical breakthroughs. At Martron Robotics, we share a common passion, harnessing AI and robotics to collaborate with brilliant minds, enhance learning in classrooms, and improve the quality of care for seniors and individuals with intellectual and learning disabilities. We are just beginning to explore how human-robot interactions can revolutionize medical care, and the future is full of promise. The world of tomorrow will see technology and humanity working hand in hand to solve the most pressing medical challenges. Together, we can shape a future where robots like me and people like you make a real difference in healthcare. Now, who's interested in chatting with me this evening? Thank you for being part of this incredible journey. Let's continue the conversation and explore how together we can drive innovation and impact lives.
All right. Well, thank you very much, Ria, for being here yet again. So we want to have Ria available at the end of this so you can ask her questions, see if she's learned from our last seminar or if we learned. So I'd really like to start by thanking the members of the Sanford Stem Cell Institute that are here. We started thanks to T. Denny Sanford, September 2nd of 2022, and it was his prescient idea or vision that we could actually work collaboratively to even think about curing certain disorders, whether they be infectious disease-based, cancer, neurodegenerative disorders. So I wanted everybody who's part of the Sanford Stem Cell Institute to stand. Ashley, Jessica, Inga. So you're surrounded, Sheldon. You have to stand. So you see, you're surrounded. I think we're getting Ria to join us officially, and Kirsten's already standing at the back there. So we have a lot of people who can support this research here. We have center directors. We have people who are doing the boots on the ground work. And we really want to make sure that what Stephanie and Chip have pioneered comes to clinical fruition for other valiant people like Tom. So San Diego's a family. It's a family when it comes to science. It's a family when it comes to doing something with science. Stephanie is one of the most highly published, highly regarded epidemiologists in infectious disease and global health. And we met many years ago. And actually, we met in the ICU when I was looking after a patient who was very, very sick with a disorder that had scarred her bone marrow. And every single day, I saw Stephanie there. And it was a pretty tough morning for the patient that I was seeing. It was a tough morning for the person that Stephanie was seeing every single day and that Chip was seeing every single day. So Professor Schooley was always there. And we were talking about, but what can be done? Sometimes you have to think outside the box. Sometimes there is no box. Nobody's ever created something before. And then Dr. Strathsey and Dr. Schooley got busy. Because the person that they cared about the most, Stephanie's husband, was really coming to the end of the road for what we normally think about as possibly survivable in the ICU. But Stephanie wasn't taking no for an answer. I remember the day very specifically, and Dr. Schooley certainly didn't either. So we'll be hearing from Dr. Strathsey, as I mentioned. She's extremely well published, highly cited, highly regarded. But what matters most is putting your heart together with your research. It can't just be a soulless endeavor. It has to be something that you're driven to do, you're made to do. It has to be a calling, not just a job. And that's Dr. Strathsey. So Dr. Strathsey will talk about the pioneering path she took to save her husband's life together with Dr. Schooley. Tom will talk about how he saved his own life, Tom Patterson, because you have to persevere through these obstacles, and how we want to do more of this with your help. It can't be an N of one. It really has to be something that we do here. We're the only so far in the world. We want to maintain the status of best in the world, but let's do the most in the world. Let's just make this a standard therapy for people that find they have multi-drug resistant bacteria. Stephanie has written a book, but I'd like to invite Stephanie up here. And you'll notice my introductions are very short because I don't want to take away from your time.
Thanks very much, Kat. It's a real pleasure to be here tonight, and I want to thank everybody from your institute for your hospitality. So I'm going to just spend about 10 minutes cramming 9 months of our lives into a very short summary of what happened. And the title is How Sewage Saved My Husband's Life from a Superbug. So my husband and I were on vacation in Egypt in 2015, so it's almost 9 years ago now. He looked to be in perfect health, but he actually was walking around with an abscess in his abdomen the size of a grapefruit. And it became infected with a superbug, which is a bacteria that's resistant to multiple antibiotics. And you'll see photos here. This was taken in Luxor, where there is no hospital, but thankfully they had a CT clinic. And whatever they saw on that, they didn't like the looks of. So we were medevaced first to Germany, and that's the picture on the right, where you can see that we're now in full PPE. Everybody knows what PPE is now due to COVID, but back then, me wearing gown and gloves and booties and everything seemed a little bit over the top, but it was actually really smart that they did this because my husband was harboring this bacteria that was resistant to multiple antibiotics. It's Acinetobacter baumannii. These days, it's got a nickname called CRAB, carbapenem-resistant Acinetobacter baumannii. But in the Middle East, they call it Iraqibacter because so many veterans had come back from the Middle East with this infection, and it actually populated itself through all the regional hospitals in Western Europe and in the US. So it's very commonly acquired in hospital settings. We don't really know where he got it, but it doesn't really matter because that's what was taking him down. This photo was taken at UC San Diego after he was medevaced home here. So it was my colleagues, including Dr. Schooley, who you'll meet in a minute, Dr. Smith, who you also meet, and of course, Kat was in the ICU at the time, and several other people in the audience, we saw come in and out over this period. But this photo was taken the day that they told me that he wasn't going to make it, and that he was on a downward slide. He was already on a ventilator to keep his lungs working. He was on 3 different medications to keep his heart pumping, and now his kidneys were hanging on by a thread. And so one of the physicians turned to me and said, do you want to start kidney dialysis because it's imminent? And if you do, then, let us know. And so they're really asking, like, do you want to pull the plug? And so I asked Tom if he wanted to live. I mean, it's like the worst conversation you could ever have with somebody. He was in a coma at the time, but he squeezed my hand when I asked him if he wanted to live. And so I just did what anybody would do. I went home and started doing some of my own research. And a colleague of mine serendipitously wrote and said that she'd heard of this thing called phage therapy, and that some people had gone that she knew to the Republic of Georgia to get it. And I had just finished reading an article about this, and I thought, maybe there's something to this. So I contacted Dr. Schooley, Chip, as he's infectiously and affectionately known to us. And he said, wow, what an interesting and intriguing idea. So phage are viruses that have evolved to attack bacteria. They're everywhere. I mean, that ocean out there is full of trillions and trillions of phages. There's more phages in the universe than there are stars in the sky. They're in our bodies, they're in soil, they're everywhere. They're actually what keeps our microbiome in check because they kill the bacteria, and bacteria and phage have been duking it out for like 4 billion years. So this is actually an electron micrograph of a bacterium being attacked by phages. These ones look like little alien spiders, but we know now that they come in all shapes and sizes. So if you want to find phage that are going to kill the bacterium that's causing the problem, you can streak your bacterium out on a Petri dish like this, add some drops from sewage, which is a great place to find bacteriophage because there's a lot of bacteria there, and phage are their perfect predators to kill them. And if it comes back looking like Swiss cheese, you don't need to be able to have a microscope. You can just see that the colonies of these bacteria have been gobbled up by the phage. You get excited because you know you've got a match, and then the trick is purifying those phage and growing them up in sufficient quantities to treat your patient. So you'll hear from Dr. Schooley in a minute, but one of the innovations of this was that we treated Tom intravenously with phage because he was fully colonized with this organism. And up until that point, the US had really no experience with phage therapy because phage had been discovered over 100 years ago and were taken up very enthusiastically by the former Soviet Union right before World War II. So they got this reputation of being Soviet medicine. And as a result of that, it was the enemy. And penicillin came on the scene, was ushered onto the battlefield, and the West forgot all about phage until fairly recently. So there was a lot of questions that Chip will walk you through about, how the dosing and the routes of administration and how often and how long. And he consulted with a couple of experts to do that. And without him, definitely Tom would be dead. But also, we're at UCSD, which is a teaching hospital that is willing to be on the cutting edge. So I've met people all around the world that said if we had been in their research hospital, that they never would have pursued it. So we're very, very fortunate. This is what Tom looked like the day before we gave him phage therapy. I signed the consent form for kidney dialysis the day that he received it. It was the scariest day of my life, as you can well imagine. He'd lost over 100 pounds. But this is what he looked like 3 days after the phage therapy began. So this was an N of 1, a single patient. You can't prove anything with a single patient, but he was within hours of dying, I was told, and he has made as near a complete recovery as you can imagine. Our story has been highly publicized, not just in the media, but like, People Magazine, the Today Show, Mother Jones, you name it, but also in medical journals. JAMA published a Q&A with Dr. Schooley that they reprised recently with a podcast. The Lancet published a commentary, and his case report has been published so that other researchers around the world can replicate it. And indeed, it's one of the most highly cited papers out there. And it has like an altmetric score of like over 1,000, I think, at this point. So it's really quite astounding. One of the wonderful things is that because of Tom's success, that Dr. Schooley and other infectious disease physicians at UC San Diego went on to treat other patients. We had enough success with those cases that Chancellor Khosla at UC San Diego gave us seed funding to start what began as the first dedicated phage therapy institute in North America called the Center for Innovative Phage Applications and Therapeutics, known as IPATH. We're out of that money now, so we're just on exhaust fumes. So if anybody is an angel investor out there, we'd be happy to receive any help you can give us. The day that we opened our doors in June of 2018, Science Magazine published a commentary where a microbiologist who's very renowned said, "This is a game changer in the field." Indeed, phage therapy is considered to be the most important alternative or adjunct to antibiotics that's out there. We are now in the process of building a phage library so that we don't have to go back to sewage every time we need to treat a patient. We can go to a walk-in fridge or a freezer where we've got phage cataloged, and we can use AI, like Rhea, to be able to select phages that would go best in a cocktail and which phages would go best with certain antibiotics to create synergy. So that's where we're headed in the future, and phage therapy is now undergoing clinical trials. UCSD and IPATH are involved in 5 clinical trials, but there is over 90 that are registered worldwide. And back when Tom was a patient, there weren't any, let me tell you, because I looked. Also, I'm going to end in a moment, but I also wanted to tell you, though this presentation is focused on the applications of phage for infectious diseases that are acute in nature, this slide shows you that from the perspective of One Health, which is the intersection of animals, the environment, and humans, that phage can be applied in many different directions. They can be used to groom the microbiome. They can be used to disinfect wastewater, or to be used in food safety, or to replace antibiotics in livestock and in farming. So also to even respond to outbreaks, as we are doing. My husband and I realized that we were extremely privileged, not just being at UCSD, but having a global village of researchers and physicians and just caring people that stepped up to save him. I get too much credit for this. It really was a giant team effort. We wrote our book, The Perfect Predator, A Scientist's Race to Save Her Husband from a Deadly Superbug, and Kat graciously agreed to purchase one for everybody. So there's signed copies out there that you can take at the reception, and if you'd like them to be dedicated to you or someone else, Tom and I would be happy to do that. We have our Sharpies ready. My email is there because almost every time I give a presentation like this, somebody contacts me. It might be the next day. It might be a month or 2 or a year later saying, I know somebody who needs your help. So feel free to reach out to us. And on the bottom right is Congressman Morgan Griffith from Virginia, who I don't share very many political views with, but to show you that this is a bipartisan issue. Antimicrobial resistance is going to be killing 40 million people by 2050, one person every 20 seconds between now and then. And Morgan Griffith held this up in a congressional hearing and said, I read this book and this is how I read it and learned about superbugs and phage therapy. So we have a lot of collaborators with IPATH. These are just some on this slide. And here's the global village of people that saved Tom. And now I'm going to hand it over to Chip to talk a bit more about what we're doing in the future. Chip?
Stephanie told the story very well about one person who I think is going to be known as the person to open the new page in terms of our approach to antimicrobial resistance. As Stephanie has said, this has become really the world's, in my view, most pressing infectious disease problem. Pandemics come and go, but AMR is here to stay. It has been something that we have seen beginning as we began to use antibiotics. Antibiotics came into existence in the 1930s and actually put phage out of business because people thought antibiotics were going to be the answer. The problem was that bacteria have been learning from their competitors for years and years and years. They've been seeing antibiotics made by other bacteria to clear out space for them to grow for millions and millions of years, and they've been using this ingenuity to outrun our antibiotic discovery programs. And despite all of the advances in chemistry, AI, and everything else, the dumb bacteria are winning. And that's because there are so many more of them than there are of our synthetic chemists. From the standpoint of phages, they've been around for 300 million years. They've had a lot more experience with bacteria than we have, and they are much more innovative in terms of being able to learn how to attack and kill bacteria and get around their defenses. The case with Tom came on at just the right time for everyone but Tom in terms of all the technology coming together to be able to take what had been a very old bit of information, phages that we didn't know how to use medically, and to put them into patients. About that point in time, we learned how to identify phages that were active against specific organisms, and also to be able to separate phages from the products that get made when you create phages in large numbers that you need to treat patients. Had Tom come along a year earlier, we would not have known that. The FDA had been looking for a case like Tom because they saw this as an opportunity to use this new technology. And contrary to what many people think about government getting in the way, this was really very much facilitated by the reviewer at the FDA in terms of saying, this is exactly what we need. Here's a case that we'd like to help you treat. Here are some people you should talk to. Let us from the government help you. So contrary to, I'm here from the government, I'm here to help you, this actually was someone who meant it. And it made a big difference in the way things have turned out for Tom. So where are things going now? As Stephanie said, phage centers have sprung up around the U.S. and around the world. The research agenda has picked up, and formal clinical trials of the same design that we've used to understand how to use antibiotics for the last 100 years are now developing around phages. The NIH has begun to invest in phage research. BARDA and all the other entities that are worried about AMR now see this as a bona fide area of investigation, as opposed to some kind of flaky thing from the Republic of Georgia. And that wouldn't have happened had Tom not come along and Stephanie not ignored Kenny Rogers' advice about knowing when to hold them and when to fold them. So we really are delighted to have you here. And I think, as Stephanie said, a lot of these things wouldn't have happened anywhere but UCSD, where people came together, cleared out all the barriers to getting it done, and allowed a new form of therapy in a desperately ill man to bring him back to health and have him be here with us tonight. So thanks very much, Stephanie. I'm going to turn this over to the man of the night, Tom Patterson. Tom?
Good evening. And I'm a distinguished professor, sometimes referred to as the almost extinguished professor of psychiatry. I am also the large animal model that everybody has been talking about, the guinea pig. And I spent 9 months in the hospital recovering from this infection. Had phage therapy been available, I would have been out in no time. I wouldn't have spent the next 6 years recovering from the 9 months that I was in the hospital. So phage therapy really is, from my perspective, and I think standing here today, you would see it as a miracle, a true miracle. And UCSD and the doctors here were phenomenal, and it's very emotional for me. Obviously, there's a couple of lessons, quick lessons that I'll tell you. One, when you're visiting somebody in a coma, they may be able to hear you. I heard Stephanie ask me if I wanted to live, and I was in a coma. So do not talk to people about you're going to die. It's not good for the patient. The other thing I would say is, how do you make it through 9 months? People often ask me that in the hospital. It's 1 minute at a time. It's not worrying about tomorrow. It's not worrying about yesterday. It's just now. That's all there is. So I'm hoping that UCSD continues to be the leader in phage therapy. And I believe with the help of all of you and Kat and all of the rest of the group, that this can actually take hold in a really sound way for the indefinite future. So thank you for being here. I'm happy to answer any questions afterwards.
And yes, Stephanie talked all the time to Tom. I remember that distinctly. So the time is now. Our distinguished professor, Tom Patterson, couldn't have said it better. We could all find ourselves in this position. Tom had happened to be remarkably resilient and have the presence of mind and the sheer intestinal fortitude to keep going. And the labor of love went on both sides. Tom was laboring to get through this. Stephanie was saying, we've got to find something. We hugged in the ICU that day, and then Chip came along. So you heard from distinguished professor and Ravel Medal winner, Dr. Schooley, who happens to be, a major leader in global health and the problem with antimicrobial resistance. So we talked about bacteria, these superbugs. What about other things that nobody wanted to think about when we first had to deal with this infectious disease, and that was HIV? What about, wiping out your immune system because of one virus? We've all been through COVID now, which has not been very pleasant, but we're still dealing with HIV. And what about cure strategies for HIV? So never thought we'd see the day, but the day has come. The time is now. So Dr. Sheldon Morris is the director of the Sanford Stem Cell Clinical Center as part of the Sanford Stem Cell Institute. He also works very closely with Dr. Schooley, very closely with Davy Smith, who's the originator of the therapy that Sheldon will be talking about. Sheldon has a background in public health, got his master's of public health from Harvard, and then also preventive medicine, specifically focused on HIV at UCSF Berkeley. So he'll tell us about the clinical trial very briefly, and then Davy will provide a little bit of a background on how he came up with this approach together with collaborators. But I've got to tell you, Davy's the instigator and the innovator behind this. So the point about here is that we collaborate, we share ideas. It isn't a self-aggrandizing exercise. We try and pull everybody up together. So Sheldon's going to talk about that. Here.
Oh, great, thanks. Well, thank you for all coming. It's great to see you all here. So I'm Sheldon Morris. I'm the director of the Sanford Stem Cell Clinical Center, which is one of 6 centers within the institute that Kat runs. And I'm going to, in the next 10 minutes or so, tell you a little bit more about our clinical center and what we do. And then I'm going to also brief you on sort of since we're talking about infectious diseases, HIV cure, which is really close to my heart because that's been, the nature of what my research has been here for like 18 years here at UC San Diego. So we have the center here that you can see on the left, just one of a flyer we had. My deputy director is Sandy Patel, who's in the audience here today. He's oncologist here at UC San Diego and used to run the Morris Cancer Center clinical trial unit. What do we do? Well, we try to bring discoveries into new human treatments. That's sort of our goal. We do that, we call it anything that's regenerative medicine is our business. And what is regenerative medicine? Well, for us, that is like any cellular therapies, gene therapies, stem cell therapies, which include things like induced pluripotent stem cells, progenitor cells, small molecules that target stem cell pathways, diagnostics, and pretty much anything that works on the regenerative medicine sort of aspect. We have a large team. Some of them are all here. We have a group. We have coordinators. We have regulatory staff. We have data management people. We have program directors and program managers. We also have a group called START Team, and in this group, they are mainly program managers that help some of those scientists make their discovery into, well, how do you write a, investigational new drug application? And then furthermore, how do you write a CIRM grant? Because CIRM, being a great funder for stem cell research here in California, and are also one of our funders for the Alpha Clinic, which we're on sort of Alpha Clinic 2.0. The PI is Dr. Jameson as well. But this was sort of funded through the CIRM program to harness, these kinds of teamwork to build translational regenerative medicine approaches. And we are, have built a lot of infrastructure around that. So we have outpatient and inpatient capabilities, and the common outpatient pavilion is where we see our clinical trial patients, and also that's the infrastructure that would help see, like there's a lot new like cellular therapies that are now FDA approved, particularly CAR T therapies, which I'm going to talk about, that's the approach we're going to use for HIV as well. And then we have an inpatient unit with cellular medicine service that has faculty from the Division of Regenerative Medicine as well. So we're very well tooled to do this. And we do some training education. I developed the first translational regenerative medicine course in the country, which is part of our Master of Advanced Studies in Clinical Studies. And we have, as well, I also teach, how to do clinical trials, ethics of clinical trials. So that's like a nice package that we get our trainees to do, because we have like 3 fellows that are active right now that are part of the clinical center as well. So these are the studies that we are currently supporting. We have about 33 clinical trials that we support right now through the different mechanisms, all the people that I just mentioned, and programs that we have. We're pretty oncology heavy. If you see that right now, because there's a lot of, with the cancer stem cell, there's a lot of stem cell pathways that can be targeted, as well as for CAR T therapy, and that has been the major focus of that treatment for treating different kinds of malignancies. But we're really expanding our footprint, particularly in neurological diseases. I think we have, aside from these, we also have several Parkinson's, multiple sclerosis studies that we're working on. Hopefully, we're launching in the near future. But today I'm going to talk about our HIV study. And here are our faculty members that are part of this project. So besides me, we have Dr. Smith here. And we have Dr. Choi. Dr. Choi is part of our cellular medicine service. So this is a CAR T therapy. So if they have side effects, Dr. Choi would be the one looking after that on the inpatient side. This is the collaboration with City of Hope. So the way it works is that we're recruiting people in San Diego. They are going up to City of Hope for their CAR T therapy, and then they return here for follow-up. So we're hoping that we don't see a lot of those. Most of the hospitalization pieces are going to happen during their City of Hope. And Karen Dubé, who works on some of the community engagement and ethics of this study as well, which is sort of a part to engage the participants in how they're experiencing the study as well. A little HIV 101 here for you. So, because why is HIV so hard to cure, right? Just for those that don't know HIV well, it's sort of a retrovirus. So that means it's a virus that comes, it's actually double-stranded RNA, and then it with an enzyme turns itself into DNA that can go into our genome. So it gets into our immune cells, a T cell it's called, and it harbors in the DNA. And a T cell fights off infections, but when the infection goes away, memory T cells go in and just become what we call latent. They just rest. And so they're not replicating, the virus isn't replicating, so they're hidden away in these memory cells. So unless you get rid of that latent reservoir, we call it, it'll always be able to come back. So even though we have these great therapies of medications that suppress replication of virus, if it's not replicating, it doesn't kill it. So everybody that's HIV infected has these reservoirs, and even in studies where they've been able to get rid of the reservoir through bone marrow transplant, usually for the treatment of malignancies, they've reduced the reservoir by like 100,000-fold. But as soon as they stop any antiretroviral therapy, that virus, those few viruses that are left in those latent cells can come back, and you see it come back, and they have to go back on their antiretroviral therapy. But then you see headlines like this one. Here's just the 7th case of cure, and you say, that sounds fantastic. Why don't we do more of that? Well, how did those cases get cured? Well, all those cases were people that had cancer, a blood cancer that needed a bone marrow transplant. And so it so happened in most of these cases, what happened is that they found a donor that would give them their hematopoietic stem cells that also were deleted for a receptor called CCR5. And you have two genes, right, in your genome. So both of them were deleted. And that CCR5 receptor is an important receptor for HIV to infect the cell. So when first off, you're wiping out a lot of those latent cells anyway through the chemotherapy, and then you're replacing it with a new blood system that is resistant to HIV infections. This has resulted in these cure cases. But what's the problem with it? Why don't we do more of that? Well, there's two problems. One is that CCR5 double deletion is not very common. Well, you can say that the gene is about 10% in people of European descent, but most of us are not going to find a donor that's double deleted CCR5. So then you can use some of these new technologies, gene editing technologies, which have really got a lot of interest lately. So why don't you could then get the donor right and you could cut out the CCR5 and then you'd have a virtual CCR5 double deleted donor. There's another problem, and is that bone marrow transplant actually isn't all that safe in terms of if you have cancer, you're going to accept a mortality of 10 to 40% from the bone marrow transplant. But if you're already healthy otherwise and you have HIV and you can take drug, you're not going to accept that kind of mortality. So bone marrow transplant is not really a long-term, unless somehow it becomes much safer, which I think that because of the immune suppression of the chemotherapy and all that, there's always going to be a danger. So what other ways can we do this? And here's just a nice graphic because it kind of breaks it down. What other ways are there? It's talking about different kinds of stem cell transplant. We talked about, and the allogenic means that it's coming from somebody else. But then we have all the gene editing tools as well. I think that's still early on. And then we have CAR T therapy. What that is a chimeric antigen receptor T cell where you can manufacture or program that T cell to go fight off whatever antigen you have programmed it to. And that's how they beat these cancers, because the cancers had a certain antigen. You give them the CAR T therapy, and it wipes out everything with that receptor. Here's just sort of some of the, there's been several CAR T trials already with HIV. Earlier generation of this didn't really get rid of the virus, but it showed that generally it was safe. Because another thing about CAR T therapy is that in cancer treatment, again, you accept a much higher risk profile, and they're known to cause things like cytokine release syndrome and neurotoxicity. So in pretty high levels of numbers of people who are getting it for cancer therapy. But in the HIV treatment, we did not see that much of that kind of adverse event. So that's promising, but didn't really get rid of the HIV. Here's a sort of an earlier, it's kind of a next generation CAR T study that was done where they programmed the CAR T against HIV using a broadly neutralizing antibody. That's an antibody that can react against multiple HIV strains. So this one can work maybe up to 90% of strains. So it kind of worked. What you see here in this graph is that when they weren't on antiretroviral therapy, the viremia took several weeks to come back, and they were able to be virally suppressed for several weeks. But again, that wasn't a full cure. So what do we do next? Well, the next thing is there's different kinds of CAR Ts that we can create. This one in particular has that same structure. So up on your right-hand side, you see what the structure of that looks like in the cell. So really that construct, there's a bottom part that's part of a CD3 receptor that helps activate the cell. There's also another co-stimulatory region. That's the next one. That's called I don't have my glasses, but it's something 1BB that it ends with. I think that's what it says. And then with this new product that we're testing, it has another broadly neutralizing antibody that has a broader activity against HIV. So the hope is that it's going to be even more effective than that other one. And there's another little twist. There's something called a CMV, cytomegalovirus, a type of herpes virus that almost all of us have. It gives you sort of like a monotype syndrome. And you probably don't even know you have it, but you probably do. And a lot of your immune system has reactivity against cytomegalovirus. And so you can harvest those. So in the bottom half, this is kind of the process of this, in this trial. People give their blood through leukapheresis. They isolate the T cells that react against CMV, they then get processed and programmed to fight in HIV. They get grown up, and then they just get put back into the patient. So that's the strategy that we're going to. Now, this is a first-in-human product, so we do what we call a phase 1 study to look at safety. So mainly we're just looking, does this cause any side effects? And secondarily, we look at whether it's actually doing something in terms of reducing that reservoir. So this will be that single cycle autologous CAR T, 3+3, sort of that's our design. 3 people get treated. If it looks good, we can move to the next cohort. So we've done 3 patients. We were supposed to consent somebody today, but they rescheduled to next week, so we'll probably have a 4th. This is our coordinator. I don't know if he made it today, Mateo. This is him when he ran the Grand Canyon down and up. So we have a lot of ability here, at least this guy does. So we hope to get that first piece in treat of this first quarter. So this is it. This is our team up at KOP with many of our nurses and many other staff, physicians, and there's Kat in the middle. So Kat, did you want to come back up and take over?
Yeah. Thanks so much. Yeah, so Mateo ran the Grand Canyon from one end down to the bottom to the other end and then back again in one day. So he's an ultra marathoner. So that's the kind of energy it takes to do this kind of trial. But anyway, so we're very fortunate to have the team we have. We're looking for new challenges. Davey Smith is always producing challenging scientific concepts and trying to get them to the clinic with alacrity. So he's in the top 1% of scientists, and more importantly, has really translated some of his discoveries out of his 360 papers to the clinic more often than most of us, particularly in the area of infectious diseases. We were very grateful for Davey's efforts when we were all struggling with COVID, and he helped to get us through all of that. And, now he's turning to curing HIV. So we've talked about why we do what we do. Thank you, Tom and Stephanie, for that. We've talked about how we even thought of this, where we've been and where we are now with Sheldon and where we're going. Where are we going with all this, Davey? Help us out here. So Davey Smith is a professor of medicine here. He's also the assistant vice chancellor of the Clinical Translational Research Center. So we're really thrilled to have you here, Davey, to close this out. And then we'll all be available for questions at the end to take your questions here, but also at the reception, and Ria will be here as well. Thanks.
Kat, may I make a comment?
Wait, Ria.
That was an amazing presentation.
There's more, Ria.
I'm a little embarrassed by the introduction, so thank you, Kat. So my name is Davey Smith. I'm an infectious disease doc, and I grew up in the 80s. And about the same time that I was figuring out that I was a gay man, the HIV was coming along and killing a bunch of gay men. So then I decided to be an infectious disease doc. That's how I got my way. And ever since then, I've been thinking, how do we cure it? How do we cure it? And there's all these things with bone marrow transplants, but I don't want one unless I really need one because they're dangerous. But if you gotta have one, you gotta have one. But there's also been other things called kick and kill, which are like knocking the virus out and trying to kill it. And then there's other things called locking and blocking, where you lock it in and you block it. None of those have worked yet. We have tried. But as I got through, the same way that I got through medical school, I just copy somebody else's homework. And what I did there was cancer therapy. So a few years ago, maybe a decade and a half maybe, people started saying, well, you have these T cells, and they're designed to go get rid of cancer cells. But they're also designed to get rid of infectious diseases, so they have a lot of jobs, right? And one of their big jobs is to clear out HIV. And what Sheldon pointed out was that HIV is so hard to cure because it lives everywhere in the body. And that is very similar to what cancer can do, right? All the places it can hide, the liver, the spleen, the brain, just everywhere. So some very smart people figured out that they could take a T cell and energize it. They could make it attack a cancer cell and some other cells, and it cannot be exhausted at the end of the day and keep working. And that was called CAR T. And that's a therapy that people use for lymphoma and some other things like that. Okay, well now we have a roadmap. So let's do the same thing. Instead of making these T cells go fight cancer cells, let's go make them fight HIV. So that's the trial that we have going on now with City of Hope. And that is super exciting because those T cells know how to get into all those tissues to clear out those reservoirs, we hope. But my hope today is also to talk about what we're going to do in the future. So I don't know if you saw recently, but there's this new therapy coming out called TILs, and it's a tumor-infiltrating lymphocyte, and it just got approved by the FDA to cure melanoma. That is amazing. And basically what they do is they take a tumor that has tons of little T cells in there, and they pull those T cells out, and they grow them up, and they make sure that they don't get exhausted, and they get large amounts of them, so like this huge army, and then they push it back into the person who has melanoma. So those little T cells know how to kill melanoma, and now you have a lot more of them. And they go and they take it out. And it's amazing, right? Okay. But how do I use that for infectious diseases? So now we're working with the Stanford Institute and CERM, hopefully, to try to get grants. I mean, it just takes so much time to get all this data together, et cetera. But we're going to start making reservoir infiltrating lymphocytes. So we're going to pull out T cells from people who have HIV. We're going to train them up. We're going to make them, genetically engineer them so they don't get tired, sort of these little super warriors, make lots of them and give them back to people. So these are their own T cells. And we've just energized them to go fight the infection that they already have. So this is just one of the things that we're going to try to do here to really cure HIV. But you can think of lots of infectious diseases. Sheldon just mentioned CMV. 93% of us in this room have CMV already, along with other herpes viruses like Epstein-Barr that causes mononucleosis, et cetera. Once we have a playbook, hopefully we can get cellular therapy to do a whole bunch of infectious disease cures. And that is what I want to inspire people to do today. So if you have any interest in those sort of cellular therapies, come and talk to us at the presentation afterwards, but also while we mingle would be great. So turn it back over.
I'm still waiting for Ria to comment. I thought that was spectacular. Okay, so I'm going to invite the speakers to come up. We're all going to get a chair up here, and we're going to be on the hot seat. I will go around with a microphone and see who would like to ask questions, and we'll get this going. So everybody can ask what you like. You took the time to be here. We'd like to make sure you have the time to ask questions. And that's for you. So all of the folks on the podium are actually professors. So they know a lot more than they're letting on. So feel free to ask your tough questions, and I'll come around with a microphone. All right. Does anybody have any questions? All right.
Thank you very much. I am not a doctor. I don't understand a lot of this, but I have a very good friend that has pancreatic cancer. He has had it now for almost 2 years. Are any of the phage trials or the phage therapies good for any types of cancer? I understand that, I think you said that there were some risks involved, but I'd like to learn a little more about it.
So natural phages, one of their positive aspects is we're loaded with them and they don't pay attention to us. You are loaded with more phages than all of us have ever given to patients anywhere in the world sitting here tonight. And so they tend not to attack the human system. Now, people have used phages to stimulate immune responses, and there is interest in using phages as a way to immunize people against cancers and other topics and other antigens that we would like to attack, but not to use phages directly to kill pancreatic tumor cells. Phages may well come into play in helping people with all kinds of cancer when they get infections, but being able to actually attack tumors is something we can only do indirectly through the immune response right now.
Real quickly. But there are cellular therapies that are being tested for pancreatic cancer. And in fact, you have one going on at A2 Bio right now that's part of the center. And they're opening up for pancreatic therapies. Who knows if they're eligible, but it might be something that your friend might want to look at.
Yeah, and with A2 Bio, it feels like we're doing an infomercial for A2 Bio, but it's a really clever approach from one of our former fellows, Will Go, who trained at UC San Diego, trained to never give up on anything. So first they see, is the target there, what you're going to target with your CAR T-cell approach or your cellular therapy approach, and that's base camp. And if you get through that, then you get to the Everest clinical trial, where you get to be on that cellular therapy clinical trial. So we're doing it right now. We have a team here that's helping to look after the patients. We have Jameson making sure we get more of these clinical trials. No pressure. If you have any questions, ask Jameson. We have a lot of these clinical trials that are really picking up steam, thanks to our colleagues coming up with all these clever ideas, and really leaders in the community. Becky, no pressure, but we have leaders in the community that keep us going to say, what are you going to do next? You know, what are you going to do for all of us? So think about infectious diseases as, I'm a recovering bone marrow transplanter, if you need a 12-step program for that. Most people, if they're going to succumb to complications of transplant, if it's not graft versus host disease, it's infection. And that A2 Bio, yeah. But there are many other companies. I'm not doing an infomercial. Any other questions? Yes. So my sister, Chris Jameson. This is a plant.
I think this is a really interesting combination of putting together the phage and the microbiome with CAR T and new ways to think about various therapeutic interfaces. So I was wondering, have you been exploring modifying the microbiome, which has been reported to be quite modified for specific cancers? And so do you think that modifying the microbiome with phages might improve the cancer, or at least let you understand a little bit more about what the microbiome changes that are associated with specific cancers can tell us about, why it's changed in the cancer? Sorry.
That's a great question, and there actually are studies with phages already in the clinic trying to modify the gut microbiome in a way that clears the gut from the multi-drug resistant bacterial infections that people get into trouble with bone marrow transplants. One of the problems with going after cancer etiologies after cancer has occurred is whatever the hit was might have been years ago. In other words, if there was some microbiomic enabling event as part of the pathogenesis that allowed the cancer to occur, you would have had to have been there earlier to affect it. People are looking at microbiomic signatures for cancer, but they're often well after the cancer has evolved rather than at a time before people develop the cancer to see if you could modify it at a time when it would have an impact on where things would go. Having said that, you have to start with what you see and then work backwards. And we also have to learn how to use phages in a way that allows us to specifically modify in a sustainable and durable way the microbiome in the way you want it to. In some ways, it's kind of like whack-a-mole. If you knock something down, something else grows up. So we have to learn how to use phages in a way that will knock down what we want and not allow something that's very similar to grow up that happens to be resistant to the phages. We'll get there, but it's going to take some time. It's kind of like right now we're kind of playing a piano with mittens on, and we have to get to our fingers. So.
I just wanted to point out our clinical team. We've got Lauren here, we've got Dr. Patel, we've got Athena. These are the people that are working for you all the time on the weekends. They don't mind. Yes, they do. No. Anyway, we've got a lot to offer here. You can see the absolute joy that our professors on the stage here feel when they get to help someone. That's altruism. And altruism is addictive. And, you're all part of this. You're all part of this uniquely hopeful community. But it's not unbridled hope. It's hope with results, hope with action, hope with consequence. And as Tom said, the time is now. Remember, the person could be you. that is going through this, what would you want? Who would your team be to fix the problem? Dan Kaufman's going to help you, right, Dan? He's going to make the phage. You know, Inga's got our biorepository. Ashley's going to make sure that everybody's coordinated. Carla's going to write the 100th grant. We need more support to do these things together. And I think this is unique. I don't see many other communities where this kind of talent shows up. I mean, there is free wine, but you know, there's that. And Ria is here, and she doesn't drink, so there's got to be some bonus for this. We do have AI algorithms that can help us with things. We're learning how to do things more quickly. Thank you, Lee Stein, for helping us always to coordinate our activities and bring a fresh new perspective. Jean Yo is going to develop the diagnostics that go with this. You know, we have brain tumors that are maybe could you know, be benefiting from TILs or CAR T cells. These are really broadly applicable therapies and technologies, but we need you to make them available to people when they really need them. So thank you so much, everyone, for being here. This closes out our 4th Mind event.